Hey everybody, today we're going to be talking about the Endospore Stain and going through how to actually run the Endospore Stain. So the Endospore Stain is a stain that's used to look for endospores. And you can think of endospores as tiny little survival capsules that certain bacteria can make. Basically, if a bacterium that can make endospores is put in an adverse environment, whether it's getting too hot or too cold, or maybe it's running out of nutrients, they can make just this tiny little capsule inside of their cell that contain just enough information to make another bacterium. Now, the reason why they do this is because endospores are incredibly sturdy, so they're resistant to pH changes, osmotic pressures, temperature changes. They can stick around for many, many years, and a lot of times they're also airborne, so they can float around and get into a more favorable environment. And in terms of the bacteria that tends to make these, a lot of times we're talking about things like the Bacillus genus and the Clostridium genus. And from a medical standpoint, we're really a little bit more concerned about the Clostridium genus because we have members such as Clostridium tetani, which can give you tetanus, or Clostridium botulinum, which can give you botulism, or things like Clostridium difficile, which can give you C. diff. So medically, again, we're kind of concerned about, some of these members and their ability to use endospores to then infect us and propagate their species. So kind of with that long intro out of the way, let's talk about the materials that you're going to need to run an endospore stain. You are going to need a hot plate with a beaker that has somewhat boiling water in it so that we can get a little bit of steam. I don't know if you guys can see that there, but there's a bit of steam coming out. So that is one of the things you're going to want to set up first to get that water boiling. You're also going to need a heat-fixed smear, so make sure that you get a smear made ahead of time. Again, get your slide, add your drop of water, put your sample in it, let it dry, heat fix it so that you're ready to go. We're also going to need our staining tray or the staining sink. And if we are going to be using the staining sink, make sure that this is set up in the sink itself so that we keep all the stains in there. You're going to need some water, some of our DI water for our rinse. You're going to need either Biblios paper or a paper towel for the blots. We're also going to need Kim wipes, and we'll talk a little bit about how we use those here in a minute. But we're going to need some Kim wipes, and I normally kind of break them down into fourths like this. It just makes them a little bit easier to work with. And it's also going to be nice if you have a pair of clothespins. That will just make this procedure a little bit easier. And then in terms of the reagents that we're going to use for this procedure, you are going to need malachite green. That is going to be our primary stain. For our Morton, we're actually going to be using the steam. So basically, the heat is going to be able to allow us to stain those endospores a little bit better. We'll talk about that more here in a minute. So our Morton is going to be heat. There's no decolorizer for this stain. And then our counter stain is going to be saffronin. So kind of with that in mind, let's start going through the steps of how we're going to do this and kind of get set up. So I'm going to take my smear, my heat-fixed smear, and I'm going to take my Kim wipe and I'm going to put that on top of it. And this is where the clothespins come into play. You can kind of put them on there to just hold that Kim wipe in place a little bit better. There we go. I'm going to take this guy and I'm going to put him up here. And I'm going to put my slide over the steam, and now I'm going to flood it with malachite green. So the reason why that I like to use the paper, and you can kind of push it down so it's down in the slide, I like using the paper. It's not required, but I like using it just so that it kind of helps keep that malachite green on there, and it helps kind of soak it into the slide a little bit better. The main thing here is that we have to do our malachite green step for 5 minutes. Basically, again, our endospores are very, very resistant and very difficult to stain because of that. So we actually have to utilize the steam and the heat to open up the cells and open up the endospores so that we can actually drive that green stain all the way into it. So that's why we have to do this for 5 minutes. Now a note on that, because we are using heat, it can dry out the slide quite a bit. So every 30 seconds or every minute or so, you're just going to want to take and just apply another drop or two of malachite green to your slide during that 5-minute duration. The way to know whether or not you've put too much stain on there is that if it starts dripping down into the beaker. So if you've done that, it's not going to ruin anything. Just kind of slow down on how much you're applying and make sure that it doesn't drip too much. Another note on that, make sure that your slide is over the middle of the beaker. If that stain starts dripping and it drips out onto the hot plate itself, it's going to go and you're going to have green specks all over your clothes. So let's try to avoid that. Now, in terms of what we would see down the microscope, if we were to look at it right now, at this point, both a endospore positive and an endospore negative cell would look completely green. But I'm going to go ahead and I'm going to assume that my 5 minutes is up just to kind of speed things up a little bit. And I'm going to take this off of here. I'm going to put it down in my staining tray. Then I'm going to go ahead and just take this paper off. So malachite green is notorious for staining really, really bad. So it's always good that if you just have a trash can sort of ready nearby so that you can put your paper in there. And that way we don't get stain all over the floor and things like that, because it stains really, really bad. Now, after I've done my primary stain, I'm going to go ahead and I'm going to rinse it. So I'm going to use my DI water. I'm going to go ahead and I'm going to give it a good rinse. And one thing that is going to be different than our normal rinses is that we're actually going to want to rinse for about 30 seconds or so. The main thing here is that because we're using steam and because we're leaving it stained for so long, the stain will basically stick to the slide quite a bit, and it's just going to be soaked down into the cells. It's going to be soaked onto the slide. So our normal like 3 to 5 second rinse is not going to be long enough. So we want to make sure we rinse for about 30 seconds or so. It gets a lot of that excess green off of there. Now, once I'm done with my rinse, I am ready to do my counter stain. I guess I should mention at this point, what we should be seeing down the microscope is that in an endospore positive organism, the cell will look transparent with a green dot in it, whereas an endospore negative organism would look just completely transparent. So that's what our counter stain is for here. So we're going to go ahead and we're going to counter stain with safranin. So flood the slide with safranin, and we will counter stain for about 30 seconds. Now the safranin again is a red stain, and this will basically stain the cells but not the endospores. So it kind of helps put a bit of contrast with the background, meaning that in an endospore positive organism, what we would see down the microscope is now going to be a red cell with a green dot in it. Whereas in an endospore negative organism, the entire cell will just look red at this point. So we're going to assume again that our 30 seconds is up. I'm just going to give it a nice quick rinse, get some of that safranin off of there. All right, I'm going to take my paper towel, and again I am going to go ahead and I am just going to blot my sample, right? I am not going to wipe it, I'm just going to go ahead and blot it down. But at this point, again, my slide is now ready to go. I can look at it under oil immersion in my microscope, and just to kind of show you guys again what that's going to look like, here's a picture of that. And again, what we're looking at here is, again, we have our red cells, and inside of that red cell we have that little green dot. So that little green dot is what you're going to be looking for in an endospore positive organism. If you just have a red cell with no green dot, that's going to be an endospore negative organism. Well, that should include everything that we need to know for the endospore